Hi, I'm Chris, and in today's lesson, I want to do a quick introduction of base pairing in DNA and RNA. So your nucleotides pair up in each, which is what holds those strands of DNA together. Here in DNA, A pairs with T and C pairs with G. The number of lines that you see there indicate the number of hydrogen bonds between those. So a DNA strand that has a lot of AT pairs is going to be a little bit less tightly connected than something with a lot of CG pairing, which means that it's easier to denature if you have a lot of ATs versus a lot of CGs. The difference with RNA as compared with DNA is that RNA is going to substitute a U in place of a T. So you have uracil instead of thymine, but the rest of it is going to work very similarly. And so in this case, what you see here is your pairings will be A double bonded to the U, or 2 hydrogen bonds connecting it to the U, and then your CG pair will have 3 hydrogen bonds connecting those, just like it does in DNA. So if we take a look, a lot of times you have a question where you'll see a sample strand and then they'll ask you what the corresponding resulting strand would be after, let's say, transcription, something like that, where you're converting the DNA into the RNA. So first we want to recognize that if you see T's anywhere in it, you know that it's DNA. So this is going to be a DNA molecule, and then we want to know what the RNA is going to look like. And the 3' and 5' distinction is extremely important. So this is a question you could see on any biology exam, an MCAT, a DAT, a PCAT, any standardized test that deals with biology. And one of the trickiest parts about it that test takers miss all the time, so I really want to make sure to emphasize this, is that the way that the strand aligns is extremely important, and the way the answer choices are listed can also be extremely important and tricky. So here, the way that the pairing works is any time you are replicating DNA, or if you're transcribing DNA into RNA, it's going to align itself anti-parallel, which means if one strand goes 3' to 5', the other strand would go 5' to 3'. So make sure, because in your answer choices, a lot of times, they'll show you the correct answer, but then they'll misalign the 5' and the 3' and change that up and cause us to miss a question that we really did know how to do in terms of the actual science behind it and the correct base pairings, but we just had a little bit of a careless mistake because we overlooked the fact that they labeled it backwards with 3' to 5' instead of 5' to 3'. So just make sure we're very careful about that. But in this case, let's say we were transcribing this DNA strand into RNA. Let's see what that RNA strand would look like. So in RNA, A pairs with, so hopefully we're looking up here and we say that A pairs with U. T pairs with, so T pairs with A. So even though it has a T for the DNA, the RNA is still just going to be the A. So that part's a little tricky for people sometimes because they say, wait, I didn't think RNA had T, and it doesn't. You want to look at what T is paired with. The A is going to recognize the T as its corresponding pair, and it's going to form those hydrogen bonds between those the same way that it would in DNA. And then here, G pairs with C, G pairs with C, C pairs with G, A pairs with U. Again, these should be U's and not T's whenever you're finding the complementary nucleotide that pairs with the A for your RNA. G pairs with C, T pairs with A pairs with U, again for RNA, C pairs with G, and T pairs with A, and this would be your resulting RNA strand for that. So it's something that a lot of people, I think, whenever they're studying for a test on this, I think sometimes because the pairs are very easy to memorize, AT, CG, AU. Seems pretty straightforward. So a lot of times I think students can overlook how careful they need to be with a concept that is seemingly so simple. If you have more questions on base pairing, please feel free to comment. Also, make sure to like and subscribe and tune in for our other videos. Thanks for watching.